The next speaker and the final speaker of our morning session today is Professor Kenichiro Itami, who comes to us from the University of Nagoya in Japan. Professor Kenichiro Itami got his BS from Kyoto University in 1994 with Professor Hisanobu Ogoshi. And then he moved on to the group of Professor Yoshihiko Ito, also in Kyoto, where he got his MS and PhD in '98. From '98 to '05, he stayed in Kyoto with Professor Junichi Yoshida. He, as an assistant professor, he became associate professor with Professor Ryoji Noyori in '05, and in 2008 he has been promoted to full professor. At this point, in addition to holding an appointment of full professor at the University of Nagoya, Professor Itami is a director of Institute of Transformative Biomolecules and a research director for JST Erato project on molecular nanocarbon. Professor Itami has received multiple awards, and I won't be able to list all of them here just because of the time limitations, but some of the most notable are the R.C. Fuson Visiting Professor at UIUC. In 2015, he has been awarded the Arthur C. Cope Scholar Award by the ACS. He has received the 2014 JSPS Prize, the 2013 Novartis Chemistry Lectureship Award, the 2013 Mukayama Award, the 2012 Gottfried Wagner Prize, the 2011 Nozoe Memorial Award for Young Organic Chemists, and 2008 Merck Banyu Lectureship Award. He is an author of more than 200 publications, and today he is going to be talking to us about materials and biology-oriented CH activation, which is really his main scientific interest. Let's welcome our speaker.
Thank you very much for a very kind introduction. It is my great honor and pleasure to be participating in this very important and historical event of this American Chemical Society, NOS. So I would like to start by thanking Marisa for inviting me to this nice conference, and all the organizers, particular students, for a very wonderful organization. And also, echoing the previous speakers, I would like to congratulate Professor Larry Overman for Roger Edmonds Award. And then he has been the previous gold medalist of the Nagoya Medal of Organic Chemistry, which is a very prestigious award in Japan. And then this is the word from all Nagoya chemists. So once again, congratulations. So moving on to my lecture, let me start with the goal. The goal of our research group is to contribute developing transformative molecules. So I want to contribute to that. That is the innovative new molecules with new superb function that help solving urgent problems in science and technology, and hopefully to contribute and change the way that we live. Of course, this is not that easy, but I hope in some way we will be able to contribute to that. And having such kind of a dream in our mind, our group has been doing lots of catalysis for materials and biology. So mainly focus is this aryl assembled molecule, which is very simple, but it produced a number of previous transformative molecules, such as drugs, such as materials, such as catalysis. So what we do in the methodology part is to try to access these molecular entities as rapid as possible. So we're basically developing new reactions or new catalysts, mainly focusing on CH activation. We not only make molecule, but we want to see our molecule working. So there are several different kinds of applications. First direction is try to develop small molecule that related to pharmaceuticals or plant animal biologies. So other direction goes to related to materials. For example, carbon nanotubes or graphenes. So these are, in my eye, extreme example of aryl assembled molecule. And what we try to do in this program is to make this entity as a single molecule to do lots of interesting stuff. So today I would like to show some brief overview as well as the recent achievement in our group. So here I'd like to summarize our CH coupling catalyst for the last 10 years in Nagoya. So as I said, our group is primarily focusing on anion assembled molecules, so the target reaction is very simple. We try to activate the CH bond of aromatics or heterocycles and try to couple that with an appropriate coupling partner to make this in a single step. But if you talk about aromatic, there are many different kinds. For example, biology field or pharmaceutical scientists are interested in a heterocycle containing aromatic heterocycles. Or people in the materials field are interested in a polycyclic aromatic hydrocarbons. There are a difference, but we developed the catalyst or reaction based on this electronic or reactivity of these kind of molecules. So for the last 10 years, we were able to develop catalysts activating and transforming almost all CH bond that is shown here by catalyst control, not by directing groups. But more importantly, we were able to develop unique catalysts activating otherwise unreactive CH bond that are highlighted in color that is shown here by unique rhodium or platinum-based catalyst that is shown here. For example, it is not easy to hit the beta position of pyrroles or thiophene, but our catalyst can do that. So this is the power of catalysts. And luckily, some catalysts became commercially available, as exemplified by this nickel catalyst. We introduced the first aromatic CH relation using nickel in 2009, but these days, these diphosphine-based catalysts turn out not only active, can facilitate activation of CH bond, but also CO bond. But there are some important aromatics lacking here. So for example, like a benzene or pyridine, which is a #1 and #2 aromatic used in pharmaceutical, this was very, very hard target for us. But very recently, we finally found some solution for activating CH bond. But today, I'm not going to tell you about all the reactions or catalysts that we developed here, but we rather would like to tell you about what we can do with this catalyst later on in my lecture. More recently, we decided to explore our methodology campaign beyond the aromatic CH bond relation chemistry. For example, very recently we found there's some very sterically hindered unique copper-based catalysts can catalyze putting nitrogen or amidation of the functional aromatic groups. So this is a very, very exciting field in our group. But today I would like to talk about these two topics. So in my group, we have a very, very simple but long sought after catalytic reaction. That is per-selective functionalization of benzene ring in a general manner. Of course, we all know that if you have an aniline derivative, you can do the aromatic electrophilic substitution reaction in a per position, but we don't have this toolbox in a general way. Per-selective functionalization, whatever you have at the position right here. I know that if we develop this, it will have a huge impact in the field of biomolecules and materials because of the following reason. For example, once you found some biologically active lead compounds, like in the pharmaceutical relevant molecules, mostly it contains phenyl groups or benzene ring. And what people do in the medicinal chemists, for example, is to try to do the per functionalization or derivatization, because in our body, in our cell, the per position of benzene ring is known to undergo the P450 catalyzed oxidation. So we wanted to put lots of different type of substituent to increase the metabolic stability or increasing the activity and selectivity. This is same in the field of optoelectronic compounds. The per position in the benzene ring is the #1 position that you can tune the optoelectronic property of the parent compound. But because of the lack of this reaction, this is time consuming. Or many cases, you have to go all the way back to the starting materials and then to put the per substituent and do the synthesis again and again. But if you have this, you can do the late stage functionalization of these compounds easily. So we started this campaign in 2009, starting from our favorite CH relation chemistry, but unfortunately, we do not have success yet, this one. So we decided to change our strategy to some extent. What about iridium catalyzed borylation followed by functionalization. It took 2 steps, but there are a bunch of organoboron functionalization out there, so this may be very, very useful. But there's a problem here. We all know that the typical iridium catalyst cannot distinguish para-CH bond and meta-CH bond, which result in the random CH borylation at this 3-CH bond forming the meta and para favoring the meta isomer. This has been the golden rule. As already said, if you use a typical bipuryl-based iridium catalyst, you cannot distinguish usually the meta-CH approach and para-CH approach. So we thought naively, what about this? What about if you have an asterically hindered ligand on iridium? Maybe there's a chance to block the meta-CH approach to form the para-isomer preferentially. So this has been our approach for the last 3 years. So my very capable grad student, Yutaro, extensively synthesized lots of different kinds of hindered ligand, more than 200, and I'm not going to tell about all of them, but this is his final answer. So he found that this particular xylomethoxy bifid ligand is effective. And if you run the reaction in hexane, you have a hyper-selective CH borylation of monosubstituted benzene. And here's an example. This is a per product, and this is shown in black is here is per selectivity, and this is a total yield of CH borylation. As you can see here, if you have a reasonably big substituent, then you have a very high per selectivity. You can tolerate some esters, free hydroxy, solid ether, or ketals in this reaction. So this is very, very high, highest. But I had to admit that this is not our final answer because there's a limitation as well. Obviously, these pair selectivities come from the steric repulsion between the substituent on the benzene ring and the substituent on the ligand. So if you decrease the sterics of the substituent, we obviously see the decrease in the pair selectivity. So we're not perfect yet, but we came to this point. So what we can do is, for example, a late-stage drug functionalization. So here's one example. The coramifan, which is a therapeutic agent for Parkinson's disease, and then Yutaro took these drugs and then subjected to his CH borylation condition, and he obtained this para-borylated product in a 61% isolated yield. And this is going to be the common intermediate for the further functionalization. Following the method that is developed by Professor Hartwig or Professor Suzuki, Miyaura, using the copper-mediated or plating method, you can install this changes to halogen, nitride, hydroxy group, triethyl methyl group, or a range of aryl group. And each derivative was made in one step from the common intermediate. So if you wanted to make n different types of derivative, you can only have to do is n+1 steps for making that. If you're doing the previous synthesis, it's going to be like 5n steps. So this is a very huge advantage of making a lot of derivatives. So let me summarize Yutaro's catalyst. So as I already said, if you use a standard bipuryl ligands, CH borylation typically goes to meta-rich. But if you use his ligand, you can change the regional selectivity completely. But I have to admit or say that independent to our work, Professor Hartwig, which is a pioneer in this field, reported the regional selective per-rich rhodium catalyzed CH acetylation reaction in Science 2014. There's a difference between iridium and rhodium and the borylation and acetylation, but I think in the mechanistically it shares a lot in common here. And also, we're not perfect yet, so I think it's mechanistic study as well as the ligand development of truly efficient ligand to form a perfectly per-selective is currently maybe the clear direction from now on. Okay, let me switch gear to the second topic. As already mentioned, the organoboron compound is very useful for making lots of different types of derivative. For example, like Suzuki cross-coupling reaction. So in a Suzuki cross-coupling reaction, You couple electrophile with the boric acid in the presence of palladium or nickel catalyst. We all know that. Typically, organohalogen compound has been used as electrophile, but recently, nitrogen, oxygen, and sulfur-based electrophile has been also developed. But what I'd like to introduce today is something a little different and unique. My grad student, Kei Muto, found that you can use phenyl ester as an electrophile. Phenyl ester, this moiety is now behaving as a leaving group in the Suzuki type cross-coupling reaction. The advantage of using ester is obvious because esters or carboxylic acids are everywhere and many are commercially available. And also, we all know that esters are often needed in the heterocycle synthesis. As you can see here, esters are needed. If you wanted to put something right here, people have been doing hydrolysis, decarboxylation, bromination, and put something on it. But if you have an ester cross-coupling, you can use this functional group directly as a leaving group. So this is an obvious advantage. I won't go for details about how he developed this, but let me go for conclusion. The K found the very inexpensive catalytic condition that is shown here. Catalyst precursors, the nickel acetate, which is our favorite since 2009, which is 300 times cheaper than palladium acetate. Butyrophosphine, this is also very inexpensive, and we don't need any base, but sodium carbonate helps. And toluene 150 degree, if under these conditions, a range of phenyl ester coupled with their boric acid. Here I would like to show the scope of ester. I'm sorry that this is so small, but in essence, a range of aryl, heteroaryls, or alkyl groups can be incorporated. The only limitation here is maybe the 2-pyridyl series. So this has to be improved later on. Another interesting aspect is this is very selective to phenyl ester. As you can see here, methyl ester or other esters are not reactive at all. So here's a scope of boronic acid. As you can see here, aryl, heteroaryl, alkyl group can be installed. Big aromatics or a sterically hindered one is no problem here. So what about aliphatic coupling, sp3 coupling? There's a limitation. We can currently only use the benzylic phenyl ester as electrophile, and also we have to add DMAP as a co-catalyst, even though it's not high yielding. So there's a room for improvement in the future. So here's a mechanism that is supported by the calculation by Jamal and Emory University. So basically we think that this is nickel 0, nickel 2 redox catalysis. Start from nickel 0, the first step is this oxidative addition that the acyl O bond right here. But before that, we found that there's a pi complexation with nickel, with a carbonyl, as well as the phenyl group pi groups. I think this pi coordination is a key for why phenyl ester is special for that. And then the second step is the transmetallation with the boronic acid. You don't need a base, but if you have a base, you form some cluster. Rate determining step is the following decarbonylation step, and then final step is the reductive elimination. So today I'm going to skip all this synthetic applications and so on, and orthogonal coupling, but I think this ester cross-coupling may be a very useful alternative in making or connecting molecules. Maybe to some extent, maybe you can change the way we think how to make molecules. We not only develop reactions or catalysts, but we also do lots of synthesis. So here's a kind of selected example that we made, bioactive natural product in pharmaceuticals by using CH coupling reaction that we developed here. And then some of their catalysts were modified and then applied to the ton scale production of the real pharmaceutical at the pharmaceutical company, which also highlights the power of CH activation. But today I'm not going to tell you about all of these examples, none of these examples, because I'm not Professor Larry Overman, I'm not Mo Mobasagi, so there's an obvious limitation in the structure that we can create it. But I have a question. Are we unique in this type of chemistry. There are many, many natural product synthesis, pharmaceutical, medicinal chemist, CH activation. All the keywords are very, very known, right? So the question is our synthesis and reaction otherwise impossible or difficult? Maybe we streamline some synthesis, but it's not otherwise impossible, we think. Or another question, are we generating new molecules and new functions? Maybe not in this particular case, because natural products were made by nature. All the pharmaceutical compounds are made by medicinal chemists already. So we only provided the streamlined synthesis or artificial synthesis. So we obviously need to do something. So the best way to do is to explore for new directions or unconventional direction other than pharmaceutical science or natural product with new partners to show the power of molecule and synthesis. So actually this was the greatest motivation why I decided to establish new research institute at Nagoya University, Institute of Transformative Biomolecule, ITBM. This is 2 years old, very young, but very exciting international interdisciplinary research institute of chemistry and biology. What we decided to do is to merge synthetic chemistry with plant biology, animal biology, and theoretical chemistry. And these are the 11 founding PIs of our institute, in addition to 7 Nagoya PIs. We have 4 overseas PIs, including Jeff Bodie from ETH. So this is extremely, extremely exciting. So our new building just completed like this, and we designed this new research building for effective merging of chemistry and biology and many people. So maybe if you have some time to come to Nagoya, please visit us. This is very, very exciting, I promise you. So obviously, after this new research institute established, we started the new project. Our role in an ITBM is very clear. Apply cutting-edge synthesis or catalysis or CH activation toward developing transformative biomolecules. In my group, we have 3 focus: plant growth, biological clock, and bioimaging. For example, plant growth and development is critically important for the global food crisis, for example. What we would like to have is a small molecule that, precisely controlling those plant growth and development. And then by transforming the plant hormone or natural product by using CH activation, or by using our library-based approach using CH activation, we were able to develop or discover a number of small molecules that can do this. Bioimaging is also important. We were able to develop some small molecule that can do exciting bioimaging. So maybe this is coming soon. But today I'm going to focus, just tell you one story about this biological clock. It's heavy, okay. So as you all know, we are living in a 24-hour rhythm of day and night every day. This is very precise. This is not only for human beings, but all the animals and all plants and many, many others. 95% of biological system is having this biological clock. The reason why we can maintain this 24-hour rhythm every day very precisely is because in our cell, there exists several clock proteins, and they're making some feedback networks. So this is a reason. So what we wanted to do here is very simple. Try to develop small molecule that can interact with this clock protein so that we can change this 24-hour rhythm forward or backward. If we can do so, such small molecule will be extremely useful as a tool to understand clock mechanism, which is not that easy, clear yet. And also maybe you have there's a potential use of the drugs for various diseases, such as metabolic diseases, sleep disorder, or jet lag, like myself now. Or in particular in an ITBM, we're interested in improving animal production or change of plant flowering time because it has been known that this is closely related within a biological clock. So today, I would like to tell about one story, recent story, about the small molecule modulator of mammalian biological clock. In a mammalian system, there are 4 clock proteins, only 4, and their function is very simple as well, which is clock, BMA1, CRY, and PER. What this clock and BML1 is doing is actually these are the activator for a transcription factor for generating other two proteins, cry and purr. Such generated two proteins forms dimer, and this heterodimer is functioning as an inhibitor of this transcription. So this is an activator, and this is inhibitor. And it takes 24 hours to go one cycle of this core feedback loop. And there's another pathway going outside of this loop. For example, cry, cryptochrome, undergoes ubiquitin-dependent degradation mediated by FBXL3. So this is a pathway to control the concentration of this guy. The breakthrough in this field happened in 2012 when Kate and Hirota, now in ITBM, discovered this carbazole molecule called KL-ZOZO1. They discovered this molecule by cell-based phenotypic screen from the chemical library. And they discovered that this is targeting cryptochrome, cry. And the mode of action is very simple as well. The KL-ZOZO1 binds to cry to stabilize toward degradation. And that means that if you have a KL-ZOZO1, you're going to increase the relative concentration of cry. That means that results in an increase of relative concentration of the heterodimer, which means that you have a more inhibition. That means that you're going to lengthen the 24 hours. In 1 micromolar, if you have this, you will have an approximately 26 hours of one cycle. But when this was reported, nothing was known about the structure-activity relationship. So I asked my undergrad student at that time, Tsuyoshi, to do the first SAR and hopefully discover the new modulator using CH activation, if possible. So he would like to summarize his undergrad study as SAR. So what he found that this carbazole part is essential for activity. If you replace this with a diphenylamines or phenylendo, you completely lose activity, which means that this is essential. This amino alcohol part shown in white was not that critical for activity, including the chiral center that is shown here. The furan part can be replaced with other substituent, which we thought that there's an opportunity. Because this is heterocycle, we have a catalyst that can activate in a regioselective manner. Maybe we can use this technology for the late-stage diversification. But furan is not fun substrate to work with. So what he did first is to replace this furan to thiophan. And now it maintained the Pier's lengthened activity. So now we can hit the alpha CH bond and the beta CH bond as well using our catalyst. And here's one example. By using our previously reported beta arylation, selective arylation catalyst, platinum acetate, butyryl tempo, and the boronic acid system, you can put the range of air group right here. Some of them show period shortening effect, which was very surprising because this is the first period shortening molecule targeting cryptochrome. So we're so excited and then did lots of biological testing. So for example, this is an original molecule and this is our new molecule. If you have KL-ZOZO1, as I already showed you, it has a period lengthening effect. New molecule induced a period shortening effect. If you have the two molecules at the same time, you go to the middle. It looks like sharing the same binding pocket, but the cryptochrome. It is also suggested from the docking study. Shown in pink is this KL-ZOZO1 original molecule. Shown in green is this new molecule, and it looks like it's sharing the same binding pocket. We spent another 1 year to explore why this is happening, why this is inducing pure shortening effect, why this is going for the opposite effect. But unfortunately, we don't have that yet. But we learned a lot of things, and we can clearly say two things. So CH activation did two things. First, and unveil an untapped chemical space for biological importance, which is right here. And also, CH activation helped us discover the first period shortening molecule targeting cryptochrome, which was lucky for us. We have another different types of small molecule that can change the rhythm of mammalian biological clock, but we learned a lot. We learned that a cutting-edge synthesis or catalysis or CH activation can contribute to the cutting-edge, real hot topic in biology. So this is something that we learned. So I hope that I was able to convince you that CH activation can impact the biology field. But other question is it also the case for material science? The answer is yes. And here I would like to tell a little story about related to nanocarbon. So from the discovery of fullerene in 1985, and followed by the discovery of carbon nanotubes, and followed by isolation of graphene, nanocarbon has been the very hot materials in various ways. So what we want to do in this field is the following. Try to address the grand challenge in nanocarbon science, which is related to the mixture problem. Of course, we can have carbon nanotubes or graphene now, but the problem is the current synthesis provides mixture of various structures, various diameters, various soluble structures, various edge structures, and we cannot separate them in a pure form. The problem is all different structure has different properties, which means that most of the property of nanocarbon that we know today is actually an average of this mixture. So we want to challenge this limit by making structurally uniform nanocarbon as a single molecule so that we can enjoy the molecular science here. For example, we're interested in making carbon nanotube by bottom up since 2005 when I joined Nagoya University. Our strategy is very simple. First, to make the shortest segment, that is the ring, and then grow them. It took us 4 years, but we were able to make these rings cycloprophenylene and by using the power of catalyst. I should also mention that we're not the only one that achieved this. Professor Bertozzi Jasti group, also Yamago group, and many others synthesize CPP these days. And then now a range of CPP became commercially available using our method. It took another 4 years to accomplish making the tubes, but we were able to grow these rings to carbon nanotube by treating this with an ethanol 500 degree 15 minutes. So this is a first ring to tube conversion. And then the beauty of this strategy is that the diameter of carbon nanotube can be controlled by the CPP, a template. But this is only the kind of proof of principle study. We're not completely satisfied about this because the yield is low. And the chirality control is not perfect yet. So what we are trying to do now is to change the template to the more robust the belt, or like a Larry Scott end cap. We're trying to make this and try to develop them and to grow them into the tubes like this. So these are the team that are doing this. We're almost here. I think we were able to we haven't isolated it, but we have here. And also we have a very exciting result related to the carbon nanotube synthesis. Previous example was 500 degree carbon nanotube synthesis. Actually, that was way below the standard 1000 degree synthesis. But now, in our lab, we have a minus 30 degree synthesis of carbon nanotube. I hope that I will be able to report that in this year. So maybe it's not a topic of today, but we also are interested in a two-dimensional nanocarbon that is a graphene. But in our group, we're not doing or treating with an infinite sheet of graphene, but we rather are focusing on structurally uniform nanographenes or graphene nanoribbon, because it has been known that has shown or predicted that the lengths or width or edge structure can control the band gap and other properties. Then how do you make this? Making this in a structurally uniform way, the only logical way we're doing this is to introduce the essence of organic chemistry, synthesis, obviously. And it has been clearly shown for many years by Professor Klaus Millen and Max Planck. And here we'd like to show the typical approach for making nanographene. Typically, people start from this type of small pi component and do the component assembly first, such as Diels-Alder reaction, Suzuki cross-coupling reaction, or CH activation in our case, to form the soluble polyphenylene precursor and purify it and isolate it. And the second step is to do the stitching between these phenyl rings, for example, like sacred dehydrogenation, fractional vacuum pyrolysis, or like a photocyclization. This is beautiful and predictable synthesis. What I would like to introduce you today is an alternative strategy for making or functionalizing the nanographene, which we call APEX, annullated pi extension. The approach or strategy is very similar to what we did in the carbon nanotube. Start from the small template and then grow them to the bigger one. So if you're targeting nanographene, obviously template is going to be the polycyclic aromatic hydrocarbon. So in PAH, there are several specific or interesting regions such as Bay or K or L, which is related to the armchair or zigzag edge of the final nanographene structure. What we want to do here is very simple. Try to develop the one-shot and region-selective apex reactions, such as this, or this, or this. If we can develop such strategy, that will lead to the growth of template nanographene synthesis and late-stage fine-tuning of nanographene properties is going to be possible with that. It took us a lot of time, but now we were able to develop the first generation of such kind, the key region selective apex by CH activation. So we found that the palladium catalyst can do this. So this is the final answer of our students. So all you have to do is to take PAH and mix with a silicon bridge aromatic, dibenzosalo, for example. And the catalyst is this cationic palladium-2, and we use very unique orthochlorinale as a ligand and an oxidant of this transformation, which since 2011. Under these conditions, apex reaction takes place selective at K region shown here to produce this one. Here I'd like to show the mechanism. The first step is the transmetallation of this dibenzosalo with palladium-2 forming biphenyl palladium. And the second step is a complexation, pi complexation of PAH, just followed by Heck-like insertion, second transmetallation, and reductive elimination and oxidation forming apex product, and the palladium-0 are going to be reoxidized to palladium-2 by orthochlorinale. This is very selected to K region because K region is most olefinic or least aromatic. And this is very, very predictable. There are a bunch of examples. So we decided to show, use a DFT calculation whether our scenario is correct or not. So we took this model complex, cationic phenyl palladium bound with the orthochlorinale, and treated with a PAH and a complex with the various places that I show here. And then also demonstrated the following insertion process. The DFT calculation clearly told us that the K region, C9, C10, complexation followed by insertion process is most probable. And this is optimized structure of pi complex, alkyl palladium species. This is a transitional stage for insertion process. It really looks like a Heck reaction. And other than mechanism, it turns out that this apex reaction is very versatile. For example, you can do the multiple apex. If you have a bis silicon bridge aromatic, there's a 2 reaction site. You can do the 2-to-1 apex reaction forming this type of nanographene in a radio-selective manner. If you treat with if you have a pyrene which has a 2K region, you can do 1-to-2 apex reaction forming this nanographene in a radio-selective manner and also in a gram scale. If you have a bis pyrene which has a 4K region, you can do the 1-to-4 apex reaction forming this nanographene again in a radio-selective fashion. You can do the sequential apex reaction for if you treat the pyrene with this particular bis silicon bridge biphenyl and treated this with an excess amount, you can do the 2-to-1 apex forming this nanographene, and then the K region is still remaining here so that you can do end cap that with a dibenzosalo, for example, to do the 1-to-2 apex to form this one. In this particular example, we achieved the 4 times growth of the pi system from the template in only 2 flasks. And also, if you have treated this in equimolar amount, you have a polymer. The great thing about this strategy or reaction is that you can directly use unfunctionalized pH without any perfunctionalization, such as bromination or borylation. We designed this reaction thinking about nanographenes, but we now change our mind a little bit, because there are many, many different kinds of pi conjugated molecule by many research groups for many different purposes, which looks like our substrate. So what is happening now is many people are sending their pi extended molecules to our group, and we pi extend it and give them back. So maybe if you have some molecules of interest, having K region, maybe you can send it to us. It's for free. Okay, related to nanographin, let me introduce our long target, 10-year target, which is a hexaarylbenzene, HAB. If you stitch this one by oxidation or Shor reaction, it forms hexabenzocornate, which is a nanographin precursor. So this is an interesting. But also, other than that, this is a perfect love structure and material science. So this is a nice, attractive target for synthesis. But for myself, other aspect is the huge structure diversity of this molecule is having. Maybe we can extend this to generally substituted benzene. So here's a question to the graduate student. What is the number of possible substituted benzene from n different substituents? So this is not easy, right? Because this is not a chemistry question. This is a question of mathematics or counting theory. I didn't know that. So there's a Bernstein lemma, and this is our answer, this one. So it's not easy. So maybe there's a number here. If you have 6 different substituents, possible substituted benzene's number is more than 4,000. If you have 50, it's like this. The history of organic chemistry have achieved that we can introduce more than 50 different types of substituent in a benzene ring, but clearly we're not exploiting this structure diversity, simply because we cannot make it in a selective fashion, or we cannot purify them in a pure form. So what we want to do is, can we make all possible isomers in a programmable format using single synthetic scheme? So if you start telling about this program synthesis, maybe I have to tell a little bit about my 15 years of career of chemistry, little history in my group. So I think a program synthesis is a kind of the concept beyond the classic selective organic synthesis, in my eye. So we all know that the regio and selective synthesis of densely substituted molecules is important and useful. We all know that, including asymmetric synthesis. But some cases, accessing only one particular isomer is a case, but we don't like that. We want to make all possible isomers in a programmable format using single synthetic scheme. So this is important. So 15 years ago, we started this campaign from addressing this multi-substituted olefin program. So we started from these kind of simple starting material and developed the bond selective insulation reactions and catalysts, and finally we can install the substituent here, 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 here in sequential manner to access tri and tetrasubstituted olefin. We made a library, and we were able to develop or identify or discover new fluorescence compounds, for example. After achieving this, we move on to this heterocycle chemistry, for example, like this, or this, or this. And what we did is start from reasonable compound, and by sequence of the catalyst control, regional selective CHR relation chemistry, and a cross-coupling chemistry, we were able to achieve this multiply arylated heterocycle synthesis. Other groups are also contributing in this field. But this symmetrical benzene or pyridine was very hard for us. Actually, 10 years ago, when I first joined Nagoya University, I gave this project to my first undergrad student. But obviously, this is really, really difficult. I almost gave up this project because it was so difficult. But 3 years ago, one of my best partner, Jun Yamaguchi, associate prof, came to my office and said that, Ken, I think you're very close to your target compound. You came to this point by using CH activation cross-coupling. All you have to do is transform this tiophene ring to the benzene ring, perhaps by Diels-Alder reaction. Wow, this is exciting. Of course, I noticed that there's going to be a regional selective issue here, but at least in my eye, it looks like very, very nice So I assigned undergrad student at the time, Shin, to tackle this problem, and I achieved that. So this is how we synthesize tetraaryl thiophan, for example. We reported in the first generation synthesis in 2009, but this is in a second generation. So we start from 3-methoxythiophane and did the bromination followed by Suzuki cross-coupling in one pot, installing aryl group at the C2 position first. Next catalyst is the one that I'll show you, is the palladium acetate bipyridyl tempo system, activating here to put the aryl group right here. And the palladium silver carbonate system is effective in putting remaining CH bond here. And we convert this to tetrafluoride, and then the final step is the typical Suzuki cross-coupling reaction. By doing this, we have a 5 milligram scale of the range of aryl group at the thiophane ring. Tetraaryl thiophane, unfortunately, didn't have any Diels-Alder reactivity, simply because this is too stable and aromatic. So what they decided to do is to do the monooxidation, turn on the diene reactivity, and sure enough, from this one, you can do the thermal Diels-Alder reaction within a diphenylacetylene and followed by SO elimination to access HAB within 5 different aryl groups, right here. The structure has been confirmed by extracrystal structure analysis mostly. Yields are not great here, but we don't care about this because these are the molecules that nobody has ever synthesized. This is an example within a symmetrical acetylene, and then obviously what happened if unsymmetrical acetylene, for example like this. As expected, there's no control over regional selectivity shown here. But if you limit the number of isomer into 2, you can isolate it in many cases. For example, in this particular case, we did the reduction and I put the silyl group and then make it easy to separate them. And then the structure has been confirmed by extracrystal structure. Believe it or not, this simple system, this is in its first example to isolate or characterize HAB within 6 different aryl groups, which means that HAB is largely unexplored experimentally in terms of property. So we were able to uncover the existence of substituent effect in the photophysical property of HAB from the small library that we made it. Hopefully it's going to be helpful for the future rational design of HAB. So this reaction, Diels-Alder reaction, is not only for diaryl acetylene, but you can use benzene to access substituted naphthalene, or you can use nitrile for substituted pyridine as well. Clearly, there's still an issue of regional selectivity, but I think this is an important step toward fully programmed synthesis. Before my grad student, Shin, is going to get the PhD degree, we hope to be able to develop a really fully programmed synthesis of HAB in the future. So all of these previous examples are related with the nanographing, but the planar one. So let me tell about our story exciting story, when we change our substrate from planar to non-planar PAH. We started from front. It was a simple question. What happened if you applied our catalyst to the non-planar PAH, such as a guanylane, aromatic bowl, 1/3 structure of C60? We didn't have guanylane at that time, so I sent my student to the place of Larry Scott in Boston College. There was a surprising thing happen. When Katsaki treated this with his catalyst, palladium chloronyl, and treated with an excess amount of aeroperoxine, he found that all the CH bond at the rim of guanylane, 10 CH bond, were activated and arylated to have a decarylated guanylane, which is a very interesting chiral object. Next reaction was planned reaction. So we planned to oxidize that to close up this 6-member ring by DDQ or iron chloride. He did that, but even below room temperature, he didn't observe this, but he found that over-oxidation product, which is right here. He found that he's forming the 5 additional 7-member ring right here. This was very surprising because 7-member ring cannot be planar, and this is all sp2 hybridized carbon who wants to be planar. And this is in a structure that he confirmed by the extracrystal structure analysis. And this is all sp2 carbon, but you can see here, this is grossly warped that is shown here. Most importantly, this is having a saddle-like negative curvature, which is important for having the three-dimensional nanocarbons. So we named this in the warp nanographene. Let me tell a little bit more about the structure. So it turns out that this warp nanographene is chiral. So if you see this part, you can see this as a helicity. So you have an M and P helicity. So the most stable structure that I found in X-ray is MPMPN. Obviously, you have an enantiomer here. So how is the process of going through the racemization process? It's not going entirely through the molecules, but it's rather simple. So all you have to do is flattening this biphenyl part to this part, and you will see this mirror plane, and flip it, then you'll have this one. So this M chirality changes to P chirality. And all you have to see this is to rotate 72 degree clockwise, then you will have a PPMPN, which is an enantiomer. And it takes 19 kilocarbamole to go back and forth, which means that at room temperature, unfortunately, we cannot resolve this. But maybe substituted one can do that. And then we found that in the crystal, there's a pair of enantiomer contributing a beautiful stacking of this kind of warp nanographene. This is very beautiful, I think. So this is kind of the summary of the warp nanographene. This is clearly a new form of nanocarbon, grossly warped, three-dimensional, and chiral. And then this negative curvature is created because of this extensive seven-member ring. Only 2 steps from commercially available coaniline. And the unique property is that it's not only stable, but it's very soluble to common organic solvent. Because of the warp structure, it has a very weak intermolecular interaction. Believe it or not, the T-butyl analog is even soluble to hexane. It has a relatively large homo-lumo gap because of this warp structures, and because of this interesting 5-member ring and 7-member ring alignment, it has a reversible 2-electron oxidation and reversible 3-electron reduction, which is interesting for further application, which is now ongoing. But this is an interesting structure, but a little too small in my eye. So what we wanted to do, we designed the new molecule that is shown here. We now named the super warp nanographene consisting of 260 carbons with an emphasized warp structure shown here. Inside part is a warp nanographene, but this is decorated with a hexagonal rings. So I asked my undergrad student, Kenta, can you make this? And he said, yes. All PhD student and postdoc previously denied that. But the undergrad student always helped my ambitious kind of project. Amazingly, he synthesized this molecule in only 4 steps from coaniline using CH activation. But we haven't confirmed the structure by X-ray. So this is kind of the goal of his study from now on. So because of this purely discovery, lucky discovery, we now are start explore other types of three-dimensional curved nanocarbons. So here's one example. We designed and synthesized this new three-dimensional pi-extended double helicene consisting of 58 carbons shown here. This is interesting because in an upper part and down part, these are 6 helicene substructure. It has this obviously non-planar. But in the left-hand side and right-hand side, it has a tribenzyl paralline structure, which is pretty planar. So this is a nice combination between non-planar and planar, resulting in a three-dimensional double helicene. Takao synthesized this molecule in 2 steps from naphthalene using CH activation. So using our catalyst, palladium orthochloronyl, and treated with biphenyl peroxine, you can put 4 biphenyl group on it. It's a regional selective isomers. It doesn't matter because you're going to oxidize by DDQ to have this. Unfortunately, this is short step, but it's only 2% overall yield. So he also devised the reliable Suzuki cross-coupling route starting from dihydroxynaphthalene. 5 steps, he was able to get this. Once he gets this, he realized that there's 3 isomers. So one is this twisted isomer, which has an enantiomers, and one is a meso one. The major product is this twisted one, and this meso product is a minor one. And then we calculated how the energy is, or like a possible isomerization pathway is between twisted and a meso compound. It turns out that the twisted is more stable, which explains that we get this more. But this racemization seems not to occur under the condition that we synthesize. And also, we can separate these isomers because of this high barrier. So this is PP isomers of twisted, MM isomer of twisted, and this is a meso isomer. As you can see here, this meso is a minor isomer. We separate it, and indeed it's take a CD spectra, and as you can see here, this is a really nice enantiomer. It's related to the theoretical calculation as well. And this is an extracrystal structure of twisted isomers shown here. And this is the most exciting result. As you can see here, there's a homochiral monolayers, and then it stacks each other. This is very common to the three-dimensional nanocarbon that we have been synthesizing. What we are expecting here is, by having this, we can have both inter and an intramolecular chain transfer capability. This is something that is very unique. So this is what we want to use, this type of three-dimensional pi stacking for isotropic charge transfer, in particular for developing printable and universal charge transporting materials. Of course, this is important, but depending on what you use, typically you have a different molecular design. But we're hoping that this may be a new concept for the general and universal and printable this kind of materials. And this is ongoing now. And we have a very exciting preliminary result. Okay, I'm sorry that I went this way or that way, but I hope that I was able to convince you that it's a simple chemistry, but there's a lots of things to do, or lots of things remain to be discovered. So this is, I think, is very important. We're not in the final goal yet, but I think this is a fun project. And also, other message that I wanted to deliver is the CH activation or synthetic chemistry is very nice and useful and fun, but this is not the only science for the synthetic chemist. It has a huge opportunity, I think, in advanced biology, material science, and many other, I think. So I think it's the only, we should not limit ourselves, I think. We should, think about more and what we can do or we wanted to achieve in the future. And this is a key message, I think, to the graduate student in the audience. Okay, finally, I would like to introduce the most important key players in our chemistry. The chemistry that I tell you today, of course, not done by me, of course, but done by my very capable collaborator that is shown here. So let me start by thanking all the group staffs in our group and at ITBM, Jun Yamaguchi, Shinya Hagihara, Hideto Ito, and Kei Murakami, and also the group leaders of Erato project, particular Yasutomo Segawa and others. I'm really, really grateful to be able to have many collaborating investigators, Larry Scott in Boston College and Wakamiya and Kyoto University. Also, I'm really, really lucky to be able to have a range of capable postdoc, researchers from company, technicians, grad student, undergrad student from Nagoya University, and then also a number of exchange students from abroad, particularly from NSF Center for CH Functionalization. We accept a lot of students from there. And also, I'm really happy to have a great collaborator, in particular in an ITBM and an NSF Center for CH Functionalization. Without that, we won't be able to do such interesting things. And also, I'm really, really grateful for the financial support that I got from the government. Without that, nothing was possible. And finally, thank you very much for your kind attention. Thank you very much.
Right. Let's start with the questions from the audience.
In your warped structures, where you have your 3D interdigitated stacking, have you been able to actually measure the mobility yet in both perpendicular and parallel directions?
I'm sorry, I can't hear you.
Have you been able to measure the mobility of your your last interdigitated structure, both in the perpendicular and parallel direction.
Yes, we did. But it's the original warp nanographene is actually not having a nice mobility, unfortunately, because of this stacking intermolecular interaction is a little too weak. So we redesigned, particular for maximizing the intermolecular pi stacking. So maybe we have a new molecule, so maybe that one might have. The original one is not having a good one. Thank you.
Yeah, I have a question about your arrow ester coupling, and where you show when you have the aliphatic ester, and that's with the arrow boronic acid, it did not work well. But have you tried the other way when you have, the aliphatic boronic acid and with the arrow ester? And do you know if that work?
Aliphatic coupling that so far successful is only benzylic and then phenyl ester. That's the only one that I have. If you have a purely alkyl carboxylic acid, we only see like a 5 or 6% of the product. It's very, very low yielding at this moment. Oh, boronic acid part.
Yeah.
Oh, boronic acid, we can use alkyl boronic acid as well, yes. Oh, no, I'm sorry. Alkyl is not working. I'm sorry. Yeah, I'm sorry. I was just confusing, yes.
Yes.
How does the conductivity of the warp nanographene compare to that of the planar graphene? And is there a way to tune it based on the curvature of how you're synthesizing it?
Is it the question is the difference between the warp structure and planar structure?
Yes.
So as I said, one big advantage of warp nanographene is high solubility, but of course planar one just stacks, and then you won't be able to do anything from that. So that's one advantage. Other advantage, or not, the difference is that the homolumo gap. So homolumo gap is relatively big in the warp nanographenes compared to the flat one, which opens up some photophysics, properties. For example, it's fluorescence, but the planar one is never fluorescence in a visible region. So these are the difference, I think.
But in terms of conductivity, like mobility values.
Oh, mobility value, it's different story, I think. I think we cannot compare that. We cannot go for the value of the graphenes at all. No, I don't think so, in terms of mobility.
Thank you.
Yeah.
Are there any more questions? Since there are none, let's thank our speaker once again.
Thank you very much.